Hi everyone, my name is Nural and I'm a microbiologist at the University of Technology, Sydney. Now, I'm really interested in understanding how bacteria cause infections and finding new ways to treat or prevent these infections. And that's really how I ended up researching medicinal honey. My research focuses on two main areas. The first is looking at the antibacterial activity of honey and how we can use it directly to treat infections. For example, for things like wounds and skin conditions. And that's what I'll be talking about now. The second area is looking at whether there are any benefits to eating honey, which I'll talk about next. So honey has been used very effectively as a medicine throughout all of history, and it still remains in use today, although probably not as much as we'd like to see. The reason that it's been so popular as a medicine is because honey can have broad spectrum antimicrobial activity, which means that it can kill bacteria and other microbes that cause infections. The most popular use of honey has been as a topical treatment, particularly for wounds and skin infections, and there are many records of its use today. So just one example is that there are many records from the ancient Egyptian times showing the use of honey as a wound salve, shown in this prescription down the bottom of the slide here. And the ancient Egyptians also used it in their very complex surgeries like circumcision, which is denoted in the graphic here. The main reason that honey fell out of favor as a preferred medicine was because we discovered and started using antibiotics in the 1940s. But today we have a shortage of effective treatment options for infections because we're seeing a huge increase globally in the number of antibiotic-resistant bacteria, or superbugs. And along with climate change, antibiotic resistance poses one of the biggest threats to our society today. So antibiotics are drugs that work by killing bacteria, specifically by targeting certain things. So like you can see in this cartoon here, these antibiotics are targeting that protective cell wall or the metabolism of bacteria. But bacteria are very clever and they can evolve and change much faster than we do. So they quickly learn to fight off or resist the effects of the antibiotics. And this is what's known as antibiotic resistance. It's not that the drugs simply stop working, but rather the bacteria have changed to fight off the effect of the drugs, and these antibiotic-resistant bacteria are often referred to as superbugs. So when bacteria become superbugs, they hold on to what gives them the ability to resist antibiotics in their DNA because it gives them a competitive edge in survival. They can also swap and share this DNA with other bacteria, which means that they give and take the ability to resist other antibiotics as well. And what we end up with is resistance to multiple drugs. And these bacteria continue to pass these genes on to future generations as well. So probably one of the most exciting things about honey, at least scientifically, is that bacteria don't seem to be able to develop resistance to its killer effects. We haven't had any reported cases of honey-resistant superbugs, and we can't make them resistant in the lab either. So what we do here is we treat the bacteria with increasing doses of honey or antibiotic, and the idea is to train them to become more resistant to the treatment. So in the results here, when we see that the graph deviates above that red dotted line, that's when resistance is happening. So the bacteria are becoming resistant to the treatment is no longer effective. So if we follow the black line, which is the antibiotic treatment, we can see that resistance happens within just 2 weeks. At that 15-day point, the graph deviates above the red line. When we look at the blue line, which is the honey experiment, that line never crosses the resistance threshold. So no matter how long we train the bacteria to fight off the honey, they just can't do it. We tried this with lots of different types of bacteria and lots of different types of honey, and the results are always the same. So one of the most common misconceptions is that all honey is the same, that it's just a generic product. But actually it's very complex and has over 200 components. So in the same way that different honeys can look and taste very different, they can also have very different medicinal properties. There are multiple factors that contribute to the antimicrobial activity of honey. So the first two are that honey has a low water activity, so it's very high in sugar. And just like us, microbes need water in order to survive. The other is that honey has a low pH, so it's quite acidic, and many microbes that cause infections don't like this. Now, these two factors are similar in most honeys, but just those two factors alone make up only a very small amount of the total activity of honey. So if you've got a honey with activity coming from these two components, it's unlikely to be a good substitute for a topical antibiotic in the clinic. Now, there are some honeys that have significant levels of antibacterial activity, and this comes from the production of hydrogen peroxide. This is basically like a weak bleach, and it's toxic to bacteria. This peroxide type activity varies hugely from honey type to honey type. So there are certain honeys, like those from Australia, like Jarrah honey and spotted gum, that can have very high levels of this type of activity. And it seems to be related to the floral source, but other factors also influence this type of activity. Then there are some very rare honeys with high levels of activity that go beyond this peroxide type activity. And this is also linked to the floral factors. The most famous example of this type of activity is seen in manuka honey from New Zealand, or the related manuka-like honeys from Australia. So we think it's because of all of these antimicrobial components that we probably don't see resistance by bacteria to the killing effects of honey. So manuka honey, which is mostly derived from New Zealand, is now world famous for its medicinal properties. And the main reason for this is that it has high levels of stable antibacterial activity that comes from the floral source, the manuka plant or the leptospermum plant. This activity has been linked to high levels of a chemical called methylglyoxal, or MgO. And this occurs naturally in the honey, derived from a different chemical that occurs naturally in the nectar of some leptospermum plants. So what we see is that the high levels of MgO correlate to high levels of antibacterial activity in New Zealand manuka honeys. So manuka honeys with very high levels of MgO have a very potent level of antibacterial activity. So with all of this in mind, there are lots of things that we want to learn from honey, partly driven by scientific curiosity, but also because we want honey to be a viable treatment option and for the most appropriate honey to be used in the most appropriate way. So for this talk specifically, I'm focusing on our antimicrobial related questions. So what makes honey a good antimicrobial? Why don't bacteria become resistant to honey? How broad spectrum is that antibacterial activity? What's the mode of action of honey? Can we understand more about what it does and how it kills bacteria? And if we start using honey more and more in the clinic, where can we source more medical grade honey? So there are some things that we already know about how honey works. This is from the literature. We know that honey affects many things in bacteria. So it affects many genes in bacteria that are involved in essential processes. Things like protein synthesis, things like bacterial metabolism, their ability to divide. And we've used a couple of different model organisms to figure this out. Other things that are affected by honey are the ability of bacteria to move, its ability to cause virulence or infections, and also the way that they communicate with each other. So my PhD student, Daniel, who's here in the corner, pictured here, worked on understanding more about how honey works to kill bacteria. So his project was applying modern scientific techniques, transcriptomics specifically, to look at how bacteria respond at the DNA level to honey treatment. So he did a range of approaches looking at how different genes were expressed after honey treatment, and also coupled this with other microbiology techniques as well to give a more holistic idea of what honey actually does to kill bacteria. So our experimental setup, for those of you that are interested in the technical aspects, is that we used Pseudomonas aeruginosa as our model organism. It's a really nasty bacteria that causes nasty infections, notoriously difficult to treat because it has a lot of antibiotic resistance mechanisms. And we grew this culture up and then we split it up to apply different treatments. So of course we had a no treatment control. We then treated it with manuka honey. We also treated it with some of the components of manuka honey. So we had a sugar solution or artificial honey, and that just mimics the sugars that are naturally occurring in manuka honey, just to see what the effect of sugars were. We also looked at an MgO treatment. Again, MgO is that chemical that gives manuka honey its special potent activity. And we looked at what happens when we combine that sugar solution with the MgO, so creating an artificial manuka honey, to see how that compares to whole honey. And we looked at the transcriptomic analysis there. So one of the first things that we looked at was what kind of concentrations of these different treatments do we need to stop Pseudomonas aeruginosa from growing and to kill Pseudomonas aeruginosa. So the minimum inhibitory concentration looks at what level of honey or treatment we need to slow down or stop the growth of this bacterium. And what we saw was that we only needed a 10% solution of manuka honey to stop the growth of Pseudomonas aeruginosa. and it was killed using 12% honey. So very low concentrations of honey to both stop and kill that Pseudomonas aeruginosa. When we look at the key components in honey, so that sugar solution, the MgO solution, or the sugar and MgO combined, you can see we needed much higher concentrations of these treatments. So combining the sugar and MgO helped a little bit because it reduced that concentration. But when we look at the individual components, the action of the whole honey was much more potent. So the sum of its parts didn't quite match the whole honey. And none of these key components had a killing effect on Pseudomonas. They managed to inhibit the growth, but didn't stop or kill its growth completely. So that already tells us that even though MgO might be a huge contributor to the antibacterial activity of manuka honey, it's definitely not the whole picture. So Daniel looked at the response in gene expression to these different treatments. So this is looking at all the different genes in Pseudomonas aeruginosa and whether they get upregulated or downregulated with manuka honey treatment or treatment with the sugar solution, MgO alone, or the artificial honey with MgO combined. And I won't go into all of the details of his experiments because he spent 4 years doing this, but the summary was that manuka honey induced widespread genetic changes in Pseudomonas aeruginosa. And it wasn't just affecting specific genes or single genes, it was affecting many biological processes or pathways, so multiple genes in the same pathway. These changes that we saw were not explained by the key components in manuka honey, and especially not explained by MgO alone. And you can tell that from this graph here, where we look at the patterns in the gene changes and the colors, and you can see that none of those key components match the pattern that we see in the manuka honey treatment. And most uniquely, what Daniel identified was that manuka honey seemed to be targeting something in the protective cell membrane of Pseudomonas aeruginosa. So it was doing something to destroy or disrupt that membrane, which is the first point of entry for a bacterial cell. And this hadn't been shown before. So Daniel added a few things to what we know about how honey works to kill bacteria. Firstly, we know that MgO is not the whole story when it comes to manuka honey. There are other factors at play here as well. He confirmed that all of those previously identified processes or pathways were actually affected. So things like the cell division, the ability to move, communicating with other bacteria, and DNA or protein-related transcription and synthesis. And he identified that the membrane damaging effect of manuka honey specifically, which we're now following up on more. So this is all well and good, but if we start using honey more in the clinic, where will we source new sources of this bioactive honey? And this is where our Australian Leptospermum honey project comes in. So we know that Australia has a huge resource of Leptospermum species. We have over 80 species native to Australia compared to New Zealand, which only has that one manuka species. And it's spread all around the country, as you can see in the map here. So the idea was to survey these different species and find any other bioactive ones that might be suitable for medicinal grade honey use. So we collected over 1,000 samples from beekeepers all around the country. And the first thing we did was check their MgO levels and see how this related to the antibacterial activity, that unique activity of manuka honey. And we found that same relationship where the higher the MgO level in a honey, the more active it was. So just like in the New Zealand manuka, the Australian Leptospermum honeys also had that same correlation. We also found that we had some honey species with exceptionally high levels of MgO, so really, really active honeys, levels of activity we haven't seen before. And we identified some key species that we could target in Australia to produce these really highly active honeys. When we look at the map of where we sampled these honeys from, this shows the levels of MgO that we identified in these particular regions. So we were looking not just for particular species, but also to see whether there were certain geographical hotspots that produce this active honey. So wherever we see a green, orange, or red area, that indicates that we've got an active honey or a highly active honey in those regions. And you can see that we do have some geographic hotspots where we can really target to produce these highly active manuka type honeys. We also did some work looking at the antifungal activity of Australian honey, and we worked with dermatophyte fungi, some of which we found were hypersusceptible to honey. So dermatophyte fungi cause things like tinnias, athlete's foot, and ringworm, for example. And we were really surprised to find that it was honeys with hydrogen peroxide type activity rather than these leptospermum manuka type honeys that had this really potent antifungal effect. So just to sum up all of our results, we found that there is this lack of bacterial resistance to honey, and we think it's probably due to the many antibacterial components in honey. Honey has multiple mechanisms of action when we look at its antibacterial activity, and this is actually very different to the antifungal mechanism of action, which seems to be something totally different. So there's this huge untapped potential for further use of those non-manuka medicinal honeys. And we also found that Australia is abundant in these manuka type honeys, several of which have exceptionally high levels of antibacterial activity. So to finish off, I'd just like to thank everyone that was involved in this project, our funding bodies, of course, for making this work possible, and the Australian beekeepers who always contribute samples to our studies. And if there are any questions, I'd be happy to take them. Thanks.